Well, good morning or good day, everyone, depending on what part of the world you're in. Thank you for being here today. I'm Bill Sanders. I'm the Strecker Dean of the College of Engineering here at Carnegie Mellon. Last fall, the College of Engineering kicked off a new webinar series entitled Faculty Insights, a 20-minute briefing, which we're holding every month. In this virtual series, faculty from across the College of Engineering will share their research, its impact, and a perspective for the future. Following each 20-minute presentation, attendees will have an opportunity to direct questions and comments to the speaker. Our speaker today is Charlie Ren. Charlie is an assistant professor of biomedical engineering here at CMU. He was trained as a development biologist during his graduate study, focusing on the vascular and hemopiotic systems. Moving from vascular development to vascular engineering, he joined the Laboratory for Organ Engineering and Regeneration at Massachusetts General Hospital and Harvard Medical School as a postdoc in 2012. He became an instructor in surgery at the Harvard Medical School in 2016. During that time, he developed systematic strategies for engineering functional vasculature based on decellularized organ scaffolds. He joined the faculty of the Department of Biomedical Engineering in 2017, where his research focuses on the interface of biomaterial and stem cell engineering, with the goal of providing regenerative therapeutic solutions to repair or replace damaged tissues and organs. In his lab, Charlie is developing biologically selective and chemoselective approaches to enabling extracellular matrix modularization and functionalization to boost injury repair in vivo and whole organ bioengineering in vivo. He is also working to decipher interorgan and intra-organ heterogeneity, and therefore to engineer organ-specific vasculatures. One of his main model systems is the lung. Today, Charlie will describe technologies developed at CMU for making bioengineered mini lung tissues. These laboratory tissue models will enable us to study how genetic diseases, bacterial and viral infection, as well as air pollution, affect the human lung. Charlie, thank you for being here today to share your research and insights. Before I turn things over to Charlie, please submit any questions you have via the question and answer feature at the bottom of your screen. You can do this anytime during his talk. Ginahenry, Associate Dean for Development at the College of Engineering, will be moderating the question and answer session with Charlie at the conclusion of this presentation. Charlie, please take it away.
Thank you so much, Bill, for the very kind introduction. I also want to thank Kelly and Gina for all the efforts organizing this event. Now I'm going to share my screen. So now I'll get started. So today, I'm really glad to have this opportunity to share with everyone about our ongoing research about lung tissue engineering. In particular, we're trying to generate models that we can use to study disease, to develop therapeutics. It seems like it's never been more urgent than right now, where we're still in the middle of a pandemic with a virus that's primarily affecting the respiratory system, affecting the lung. So first one, I have a disclosure. So the research I'll describe today has been filed as a provisional patent on 2 occasions. So first, I want to give a very brief background. So as we all know, the lung is a critical organ in our body. The primary function of the lung is to exchange gas. If you were yet asked, what is the largest surface of our body to interface with the environment, many of us may think about skin. But actually, if you really look into the lung carefully, you realize actually the lung is being the largest surface for our body interface with the outside world. And someone did a rough calculation saying the surface area of the internal lung is about the size of a tennis court. And then all that entire surface is interfacing with the air we inhaled, which is the external environment. And for this reason, the lung is actually a constant battlefield between our body versus the external pollution and the microbial infections, such as virus and bacteria. That's why to have a good model to study lung infection is very important. And then one thing I want to highlight is, give you a brief overview. So once our body inhale any particles, it could be pollution, virus, bacteria, or smoking, they will passage into this respiratory tract, something we call the airway, and eventually they get to this balloon-like structure for gas exchange. And then if we take a closer look at this conducting airway, you will see it's like a tubular structure. On the luminous surface, we call them apical surface. If we take another closer view of the apical surface, you'll see a group of epithelial cells lining this surface. Some of them has little hair-like structures because they're called cilia cells, and some other cell has these vesicle-like structures because those cells are going to secrete mucus. And then those mucus get moved by the cilia, which beats in synchronized manner, as you can see on the video on the bottom, so that they can move the mucus towards the mouth. So that when, from time to time, when we don't feel well, get a fever, catch a cold, in the next morning when you wake up, you may feel you can cough some mucus out of the mouth, but the mucus are actually getting produced throughout the entire lung, but the cilia move them towards the mouth. And then either a model right now, we can study this human airway model. The answer is yes. However, they are not desired. So here I'll explain why. So usually people would put this tissue model into a hydrogel to support the organoid. So once you put this tissue model into a hydrogel, the organoid would recognize the external surface interfacing with the hydrogel to be the basolateral side. That's where they get the body support. Because of so, they would turn to their internal to be the apical surface where they're going to present their cilia. So if we, at the first look of this structure versus the real human airway, we will say, wow, it looks very similar to the human airway. However, what I want to point out is, even though it looks like it's similar, but it's actually the opposite. The reason we say that is because on the upper picture is only show a cross section, but actually this is a three-dimensional cyst. So the entire apical surface is hidden inside the organoid, inside the tissue, and it become inaccessible to all the other modeling reagents that we want to test on the infection, such as bacteria, virus, or pollution. In contrast, in the real native airway, the apical surface are directly interfacing with the external environment. So that's why when we start this project, we have a hypothesis to say, or ambitious goal, either way we can reverse the polarity of this mini human lung tissue model. Either way, we can turn the apical side from facing the inside towards facing the outside. And then like I mentioned earlier, if we think the external extracellular matrix ECM support being a critical determining factor, then what if we withdraw the ECM support from the outside? And then we hope the cell would turn to their internal for ECM support because all the cell can secrete some ECM to a certain extent. And then they would present their apical surface to their exterior surface. And then the idea is pretty straightforward. And actually, this will enable us to add all kinds of respiratory threat directly to the culture, and then they would be able to interact with the lung tissue in a physiologically relevant manner. That actually worked pretty well. So when we carried out this hydrogel-free culture over 3 weeks, so we were able to generate a living organoid where you see this red color shows the cilia presented selectively to the outside, and which is surrounded by a cage of tight junctions, which really limiting other biomolecules to infiltrate the lung tissue, exactly how our lung would function. So if you look at an electron microscope picture of this organoid, the size is about 1 to 200 micrometers. You see a lot of hair-like structures. Those are the cilia. So for those who is familiar with a Star Trek movie series, you may find this organoid looks like tribal little fictional alien species in the Star Trek universe. And then we were able to monitor this organoid over the entire 3 weeks of culture. We call it in vitro lung organogenesis. So we can see that the FOXJ1, this is a biological marker staying for the identity of the cells bearing cilia. You can see those cells show up around day 7. And then at day 7, we start to see some trace of ciliogenesis, and then they further get matured over time. And eventually, we were able to quantify, we have about 80% cells of the entire tissue to be cilia cells. Again, this is a ratio very similar to what we would find in the human lung. And then when we quantify the polarity, 100% of those organoid has a apical out polarity with the cilia presented to the outside surface. And then in collaboration with the faculty at mechanical engineers here at CMU, Dr. Amir Bharati-Farimani and his student Prakash, so we were able to develop a computational pipeline where we can really quantify how much of the surface of the organoid is being covered by cilia. Again, it's a critical physiological parameter to tell us how close is our organoid model compared to the native human lung. And then we see that upon full maturation of the organoid, we have about 80% of the surface area being covered by cilia. Again, it's a feature very similar to native human airway. And then as we'll know, earlier I just gave an analogy, usually we don't feel much mucus being accumulating in our lung unless we gotta catch a cold or get a fever. And the reason when we catch a cold or get a fever, we see mucus, feel mucus in our lung is because usually those disease conditions would cause inflammation in the lung. So when there's inflammation in the lung, they would trigger a type of cell called goblet cells. The goblet cell will secrete mucus. And then, like I said earlier, the cilia cells will move the mucus. So if we look for goblet cells through a staining of mucin, the mucus component, and then we don't usually see any goblet cells in our culture, but instead we see a lot of cilia cells, which is really reflecting a healthy human airway. However, if we treat our organoid with inflammatory cytokine, interleukin 13, really mimicking lung inflammation, and we can see a dramatic upregulation of mucin producing goblet cells. At the same time, we see a reduction of the cilia-bearing cilia cells. Again, this is really mimicking the pathological condition that we see in the human lung. Then now, please allow me to get back to our initial schematic. Seems like our hypothesis works pretty well, right? So if we put the organoid into an extracellular matrix embedding culture, and then they would have their apical surface facing the inside. If we remove the external extracellular matrix support, they would have the apical surface facing the outside. And now we want to do a further test to see how reliable this theory is. So to do that, we basically put the organoid into extracellular free culture for 1 day, only 1 day, and then followed by 20 days, put the organoid back to a hydrogel supporting culture. So basically the first day we'll tell the organoid to go facing outside, and then the following 20 days, we'll instruct the organoid to face inside. Then on day 21, we'll review what decision the organoid made. And to our surprise, when we look at this organoid in day 21, 100% of organoids still adopt outside facing apical polarity, which really indicating the very first day when the organoid was put in hydrogel free culture already determines the entire polarity of the organoid, which is very stable over the entire 3 weeks of culture. When we characterize other parameters of the organoid, is very, very similar to the first batch of organoid we showed earlier, where the entire organoid has been cultured in hydrogel-free culture over the entire 3 weeks of time. And then now we'll come to a little interesting part is, so once we put this organoid in our culture over time, when the cilia start to present on the organoid, the cilia actually, they will automatically be empowered by ATP, the power source of biological systems, which is not surprising to us because you can imagine the organoid to be a boat, or maybe you can call it a submarine because it's submerged in water. When there is hundreds, if not thousands of pedals are paddling at the same time in a synchronized manner, and then the organoid or the vehicle will actually start to move around. Even though this motility is very interesting, however, we hope to have something more stable because right now the motility looks a little bit random to us. So what we decide to do is, so what if we cast this organoid inside a hydrogel, and so that the organoid will dig a little cave for themselves to live in, and then they can propel against, and maybe that would help us to stabilize the motion of this organoid. And then that's exactly what happened. So simply transfer the same organoid into a hydrogel support, you would see the entire organoid adopt a very stable rotational motion that will last for hours and days. And then I want to highlight a potential use of this system. So right now there are 2 videos being played on the upper left screen. Myself, and I guess all of you may only be able to see the second video being played. The first video, actually, there are very, very tiny movements where the yellow arrow is pointing at. In the first movie, the motion is the individual cilia beating. Because the cilia are very, very small, they're about 1 to 200 nanometer in diameter, 2 to 3 micrometer in length. So they're so tiny, so that makes them very difficult to see. However, the second video, because it's the entire organoid, it's very easy to observe. So based on this, our idea is maybe we can transform the motility analysis of cilia, which is very small, into the analysis of the organoid rotation. So by doing so, we would amplify the subject from 100 nanometers to 50 to 100 micrometer. That means a 500 to 1,000-fold improvement in spatial resolution. And also the cilia beats very fast, about 10 to 20 hertz, but the organoid rotates much slower. That means we can achieve another 10-fold improvement in temporal resolution. All of those will make it much easier to analyze cilia motility if instead we look at organoid rotation. With this idea in mind, again, we collaborate with Amir's group. So we decided to develop a time-lapse microscopy and a motion tracking computational pipeline to allow us to reliably detect the motility of the organoid. So this is the computational pipeline Amir's group developed. So basically, we'll identify the organoid from the video recording, we'll define the region of interest, then we're going to randomly assign tracking points, and then we were able to track all those tracking points motility in a real-time simultaneous manner. So this video shows you one of the tracking is ongoing, and then we were able to calculate the average motility of each organoid. And then in order for us to finally be able to claim that we don't have to look at cilia anymore, we can look at the organoid motility as a surrogate of cilia motility. We'll have to set up the correlation between cilia moving versus organoid moving. To do that, we decide to treat the organoid with the drugs. Two drugs. One drug is called EHNA. It's a chemical inhibitor of the molecular motor that powers cilia beating. The second drug is a clinical user drug called paclitaxel. It's a chemotherapy drug treating cancers. The reason paclitaxel can treat cancer because cancer cell division depends on an internal cell structure called a microtubule. However, the same microtubule are actually used as the framework of cilia, as you can see those tube-like structures. That's why there's unfortunately a side effect of this chemotherapy drug that will cause cilia beating defect. That's why patients being treated with this drug, they will be more prone to respiratory complications. So first, we test with EHNA. Because the entire culture can be done in a non-invasive manner, we were able to monitor the same organoid before and after the 2-hour treatment of EHNA. On the left-hand side, you will see a control group without treatment. On the right-hand side, you will see a treatment group. So now I'm going to play the video. As you can see, in the control group on the left-hand side, there's a moving before and after. However, in the treatment group, before treatment, the organoid rotates really well. However, after treatment, the rotation of the organoid completely stopped. So we're able to observe a dose-dependent reduction of the organoid rotation by the EHNA treatment, and we were able to correlate that with direct monitoring of cilia motility through a conventional chemograph analysis. So we actually saw a very similar effect with paclitaxel. Because paclitaxel functioned by destroying the cilia structure, it took a longer time for paclitaxel to take effect. However, we were able to observe a time-dependent reduction of organoid rotation. And by 24 hours, since the starting of the treatment, almost all the organoids stopped moving. And then again, we were able to correlate the organoid rotation phenotype with a direct monitoring of cilia motility, where we see about half the reduction of the cilia beating frequency. And lastly, we want to bring our model to be even closer to a clinical application. So actually in human clinics, there is a group of genetic disease called primary ciliary dyskinesia, PCD. It's caused by mutation of a collection of genes. Those genes are critical for cilia structure and function. In those disease, then the motile cilia either stop beating or have irregular beating motility. So the disease is primarily affecting 4 organs, affecting the lung, the ear, the brain ventricles, and as well as the fallopian tube. That's why people suffering from those disease, they are more prone to respiratory infection, middle ear infection, brain injury, as well as infertility. So our collaboration with physicians at Washington University, Dr. Stephen Brody and Dr. Amjad Horani, we were able to obtain patient-derived cells carrying a classical PCD mutation called CCDC39. And then once we got those cells into our lab, we were able to make exactly the same organoid as we have done earlier with healthy cells. And then by looking at the morphology, characterization of the organoid, they look exactly the same between the upper line PCD, the disease cells, versus the healthy cells. However, if we get closer look at the individual cilia at nanometer resolution, we can identify the PCD organoid. Actually, all their cilia has an abnormal structure. They have an extra pair of microtubules show up in the center. And then now I want to bring them to an ultimate swimming test. to see whether their cilia is moving. So our hypothesis is because in the disease cells, the cilia cannot move, then we will not be able to see an obvious rotation of the organoid as compared to the control. And then this is exactly what we see. Well, most of the control organoid can rotate very nicely, and none of the PCD organoid can rotate at all. And then this was, again, can be correlated with a direct measurement of cilia motility through kymograph analysis. So this is all the research results I would like to share today. So here I would just like to summarize. So basically what I presented today is ongoing research in our lab has developed a new airway tissue model. Instead of conventional model who has apical in polarity, we have an apical out polarity, which really mimics the anterior facing apical polarity that's seen in the human airway that allows physiologic interaction between the airway model with respiratory virus, bacteria, or pollution particles. And then now I just want to have 2 more slides to share some outlook where we see our research is going and how would that be further connected to the real world. First, actually, we have some ongoing efforts to enable large-scale production. So we expect in very near future, we would be able to produce tens of thousands of organoids at one time. And also those organoids, those tissue models can be personalized, can be patient-derived. For many of us who have done a COVID test, one of the classical COVID tests is you take a nasal swab, have a Q-tip to take a little bit of airway tissue or airway component to test the virus. Actually, the same procedure will allow us to take some cells, the stem cells of the individual lungs of every single individuals. Then we can make organoids to be personalized, patient-derived. And also we think through this model on the right-hand side, we can enable, because the apical surface is facing outside, that's where the mucus is being produced. Mucus is one of the key pathological markers of the lung conditions. So we think we can actually sample the mucus in a non-invasive manner and for real-time analysis. Conventional analysis, you would have to sacrifice the tissue in order to do the assay. In this case, you don't have to sacrifice the tissue. You can constantly sample the secretions from the lung and then do a time course analysis. And we think this can be used for testing and treatment of respiratory threats such as virus, smoking, vaping, or pollution. And lastly, because like we said earlier, there is a motility of the organoid, and we think this can potentially be used as a patient-specific drug delivery vehicles. So basically, we can make little motile vehicles out of the cells from every single patient. And then when this organoid is moving along, they will be able to travel to the deep lung. Because the organoid is constantly beating, it makes it less likely to get stuck in somewhere along the track. And then we think this can eventually be used to express or deliver drugs or any recombinant proteins that can be expressed by live cells. With all this, I would like to thank all of our collaborators, and in particular, one of our most very talented students, Piumi, who has done most of the work that I presented today. And in the end, I would like to thank our funding source. Thank you all.
Thank you, Charlie. We really appreciate you spending the time with us today to share with us your research. It's very fascinating. And I know I learned a lot, so I hope everyone on screen learned something too. My name is Ginahenry, and as Bill mentioned earlier, I'm going to be facilitating the Q&A session with our presenter this morning. So I'd like to encourage everyone who might have questions to submit those via the Q&A box. You should see a Q&A a thing that you can click at the bottom of your screen if your screen looks like my Zoom screen, and you can just submit your questions there. So Charlie, we've already had a couple of questions come in. One of the questions relates to the fact that the BME program at the undergraduate level is a dual degree program. And sometimes people are curious about why is that and why do we do it that way? So can you talk a little bit about the dual degree program at the undergraduate level and what that looks like?
Yeah, sure. I'm happy to, even though I don't think I'm the best person to comment on this, but I'll do my best. So I think, yes, so basically all of our BME undergraduate students has another degree in another usually engineering discipline, such as chemical engineering, mechanical engineering. And we have a constant discussion inside our department. And most of us actually enjoy this really well, because all our students actually, they got really comprehensive engineer training And then they were able to apply their unique expertise from either mechanical engineering, electrical engineering, and to address a biomedical question. Just like you say, from my talk today, this would not happen at all without the close collaboration with our mechanical engineering faculty. And I think one of the way I really enjoy since I came to CMU in the past 4 years is the collaboration really make a lot of exciting research happen.
Excellent. Thank you for that. I appreciate that answer. Our next question that's come in is about sort of real world implications of this work. So can you talk about how people in their day-to-day lives might see this sort of work realized?
Yeah, thank you for this question. So Tiffany, so like I provide those outlook in the last slides, I think we really need to have a very effective model to be able to test, to develop stability reagents. I think earlier we may not recognize enough how urgent to have a good model until right now we're still being hit by a coronavirus. And right now, really, we need to be able to respond to the virus very quickly, understand the mechanism of virus infection, and then to develop stability reagent. And then actually, many of the stability reagents wouldn't get developed just by 1 out of 1. Usually the pharmaceutical companies has to test hundreds, if not thousands compounds or candidates. So that we really need a large scale, high throughput screening platform. And we think the model that we just presented can be one of such a platform because really it has the tissue architecture, cellular composition, and also the polarity very similar to a native lung. That would really, and then, like I said, in the final slide, we can produce those organoids in really large scale. That would really enable us to, the next time, if there will be a next pandemic, we can respond it in a much more effective manner.
Thank you for that. Let's hope it's quite a many years before that happens. Next question is, we know that the lung is directly exposed to air, but in your model, the engineered tissue seems to be submerged in liquid. Now, would it be possible to mimic the direct air exposure in your model?
Yeah, thank you, Gina, for the question. Yes, so that's something we've been keep thinking about as well. And we do have some preliminary data, which I didn't put into this presentation today is, so once the organoid get matured, we can actually move the organoid to be something we call air-liquid interface culture. So there is a special designed filter that is permeable to water, liquid and nutrients. So the bottom of the filter will get exposed to the liquid, which keep the organoid alive. And on the top of the filter is exposed directly to the room air. And then we were able to put the organoid right on top of the filter. So we call it organoid air liquid interface culture. And then that will be possible to expose the organoid to air. And then I think this is really a critical point to further bring our model to be closer to a real life application. And then we would be able to connect this chamber to a pollution generating chamber so that we can really analyze how does pollution particles interact with the lung tissue model, again, in a physiologically relevant manner.
Very good. Thank you for that, Charlie. Just a reminder to all of our attendees today, if you do have questions that you would like to ask our presenter, please submit those through the Q&A button at the bottom of your screen, and we will get those answered as promptly as we can. And thank you for everyone who's submitting questions already. Next question, Charlie, is how long can the engineered tissue stay alive?
Yeah, that's a great question. So would be actually one potential application we're trying to move this organoid towards is it can be potentially used for environmental sensing, and then we can put it into a field to help us to recognize where there are potentially respiratory threats, so that the human wouldn't go there. So this question brings up a lot. So based on our current understanding, once the organoid being matured, they can stay alive in a laboratory setting for 2 weeks to a month. And also we recognize, just like again the pandemic teaches us, how well they can the system work in the lab is important, but however, how well the system can work in real life is also very important, especially when we try to envision any real life application. Very recently, we did an interesting test. So we take those organoids we made in the lab, put them into Eppendorf tube. It's just tiny tube, it's closed, and then has some liquid to keep the organoid hydrated, has a little bit glucose to keep the organoid fit. Then we basically put that little tube in our drawer or in the refrigerator for 7 days without touching them, without having them have another breath of air. And then 7 days later, we took those organoids out. To our surprise, all of them are still alive. All of them are still swimming. So it really shows that this is a robust little tissue that really trying to survive.
That's amazing.
Thank you.
Okay, moving on to our next question. So the model that you presented, so would this model and analysis be applicable to studying diseases in other tissues or organs besides the lung?
Yes, I think so. Even though our primary model is the lung, because that's what we primarily focus on. But like I mentioned, so as you can see, one of the major applications of this system is to study the cilia motility. So cilia is a motile structure that's present in primarily 4 organs. It's present in the lung, in the airway, also in the middle ear, help to clear the accumulations in the middle ear. Also, it's in the brain ventricles to help to move the cerebral fluid, and also it's in the fallopian tube to help to move the egg or fertilized egg towards the uterus. So therefore, in genetic disease affecting cilia, you will actually see abnormalities in all the 4 organs. So we do think the stem cells in all other organs is very similar to the stem cell in the lung. So if we can obtain other stem cells, I do think many of the research that we showed today can be applicable to study cilia in other tissues and organs.
Great, thank you for that answer. So next question, the lung tissue that you engineered has an interesting motility. Could it be used for robotics or biorobotics applications?
Yeah, thank you. That's something we've been keep thinking and brainstorming a lot. If you remember, like I showed in the very last slide, we kind of show a little bit taste of it. So we envision this organoid is like a living tissue, has a mobility. That's pretty much how you would define a robot, right? It can move around. The only element we're missing right now, and we hope to obtain in near future, is steering mechanism. So we hope to be able to steer the organoid, which is already moving towards a specific direction. There's a couple ideas that we're considering. For example, a magnetic steering mechanism. We can potentially turn ourselves to be magnetically active. And then we can use an external magnetic field. You can imagine to guide the organoid to go to a particular part of the human body or a particular part of the lung. And because this whole organoid is built with cells that can be patient-derived, so really, we can actually design a biological robot that belongs to a particular individual and to achieve certain therapeutic applications in the body.
That's amazing. Thank you so much for that, Charlie. The future is very exciting. I'm looking forward to hearing where your research goes from here.
Thank you, Jean.
Just one last call for any final questions. If anyone has any questions, last minute questions that they want to get in before we close out this session. Okay, seeing none, I'd like to thank you again, Charlie, for this amazing, thought-provoking and informative program. I learned a lot, and I hope everyone else on the webinar learned something as well. I'd like to thank everybody who joined us today for participating in this Faculty Insights series. Please know that we will be doing more of these in the upcoming months. So I hope that you'll be able to join us at a future Faculty Insights webinar. And thank you all again for joining us. And I want to wish everyone a wonderful, happy holiday season.
Thank you, everyone.